Hello, my name is Dr. Fisher from the Center for Disease Control and Prevention in Atlanta, Georgia. Me and my colleague Dr. Brown are here today to educate you on the existence of a microscopic killer known as a prion. Dr. Brown?
Thank you, Dr. Fisher. A prion is a misfolded protein that leads to a specific form of disease known as transmissible spongiform encephalopathy. This type of disease is observed in mammals including sheep, goats, cattle, mink, deer, and even humans. Dr. Fisher?
Unlike all other known infectious agents, prions do not contain nucleic acids, but rather induce the misfolding of properly folded proteins through one of few proposed mechanisms, which will be further discussed later in the presentation. All known prion diseases cause degradation of the brain and spinal cord tissues. These diseases have no known cure and have been proven to be 100% fatal in all cases.
Although transmissible spongiform encephalopathies have been identified for over a century, the infectious agent behind the diseases was not isolated and identified until 1982, when Stanley Prusiner announced the discovery of a misfolded protein responsible for the confusing diseases, a discovery that earned him the Nobel Prize in Medicine.
Prions form from the misfolding of normal protein PRP. PRP is a sialoglycoprotein found on the membranes of cells in mammalian central nervous systems. The normal PRP protein is denoted by the superscript C. The misfolded infectious form of the PRP protein is denoted by the superscript SC. After scrapie, the form of transmissible spongiform encephalopathy found in sheep.
PRPC has a few key physiological roles in the human central nervous system. It is found to be highly concentrated in not only neural and glial cells, but also in immune cells and endothelial cells. PRPC has been found to assist in cell survival through protection from apoptosis. It also assists in cell signaling, as well as cell adhesion. PRPC also possesses some distinct functions related to cell adhesion, including monocyte migration and T-cell activation.
Although chemically identical to PRPC, PRPSC possesses physical differences that alter its solubility, structure, and stability, not to mention its deadly infectious nature. The PRPSC protein contains specific lipids as well as polyglucose scaffolds. These proteins possess a high percentage of beta sheet structure as opposed to normal alpha helix formation in PRPC. Due to the presence of the beta sheet structure, the PRPSC protein forms amyloid fibers. These amyloid fibers can grow and converge to form prion plaques, which are common indicators of prion infection.
There are two common hypotheses for the mechanism of prion replication. The first of these mechanisms implements a heterodimer complex and is referred to as the heterodimer model. In this model, a PRPC intermediate is formed during normal PRPC metabolism. This intermediate links with an exogenous PRPSC protein, forming a heterodimer complex. Within this complex, the intermediate form of PRPC protein is converted into an infectious PRPSC protein through structural change to the alpha helix into beta sheets.
The second hypothesis regarding prion replication is called the fibril prion model, in which PRPC proteins are bound to the ends of PRPSC amyloid fibers and are converted to infectious PRPSC proteins. While many researchers speculate that this method of replication is implausible due to the observed exponential increase in PRPSC proteins in infected hosts, The phenomenon of fibril breakage accounts for the exponential increase within the constraints of the fibril model. Fibril breakage creates more PRPC binding sites at breakpoints by revealing more PRP-SC protein.
With all this science speak about mechanisms of replication, we've started to lose track of the implications these little prions can have on the body's normal function. The conversion of normal PRPC proteins into PRP-SC amyloid fibers doesn't go without consequence. The loss of normal PRP proteins actually accounts for the neurodegeneration observed in all transmissible spongiform encephalopathies. Normal tissue disruption from the production of amyloid fibers causes the formation of literal holes in the tissue structure, leaving a sponge-like remnant of the once healthy tissue. These holes are the result of the formation of spongiform vacuoles in neurons. Along with hole formation, neuronal cell death is a common effect of the loss of PRPC cells due to their role in apoptosis prevention.
After hearing all the terrible things that prions have in store for your brain, you may be wondering how to stay as far from them as humanly possible. The good news is that transmissible spongiform encephalopathies are some of the rarest diseases known to man. The most popular of these diseases is definitely Creutzfeldt-Jacobs disease, or CJD. This disease is a rapid form of brain damage that leads to brain decay and decrease in muscular control. There are two types of CJD, including classic CJD and variant strain CJD. Within the type classic CJD, there are two subtypes, including sporadic classic CJD, in which the cause of production of prions is unknown, and familial classic CJD, caused by the presence of an inherited prion. In the onset of CJD, symptoms of dementia are often observed, but the disease is rarely misdiagnosed as dementia due to its extremely rapid onset.
The symptoms of Creutzfeldt-Jakob disease include rapidly developing delirium, blurred vision, changes in gait, hallucinations, lack of coordination, muscle twitching, muscle stiffness, muscle jerks or seizures, nervous jumpy feelings, personality changes, profound confusion, sleepiness, and speech impairment. There are multiple modes of infection of CJD. As Dr. Fisher mentioned, there is no known infectious cause of classic CJD, but it occurs sporadically. Variant strain CJD, however, can be acquired through exposure to contaminated food products, infected donor organs, or improper disinfection of medical tools. While the prion does not possess conventional virulence factors, the fact that it is not a living virus or bacteria causes the body to ignore it as an antigen, mounting no immune response to its presence. While classic CJD cannot be transmitted through the air or physical contact, the disease can be transmitted by contact with infectious brain tissue and infected spinal fluid.
There are a few important risk factors to be considered when discussing Creutzfeldt-Jacobs disease. On average, sporadic CJD develops approximately around age 60, with genetically linked cases occurring only slightly earlier. Genetic mutation exists that leads to misfolding of proteins, causing familial cases of CJD. The mutation occurs on the PRNP gene, which is responsible for the production of the normal PRP protein. This mutation is inherited via autosomal dominance, requiring only one copy of the mutated gene to exhibit the disease. HGA supplements from human pituitary glands and dura mater grafts provide increased risk for CJD.
Variant strain Creutzfeldt-Jacobs disease is classified as a transmissible spongiform encephalopathy due to its infectious nature. It affects younger people with an average infection age of 29, as opposed to the average age of 60 for classic CJD. Variant strain CJD is transmitted through human contact with the bovine spongiform encephalopathy agent, which causes mad cow disease in cattle. Most likely the mode of infection is through ingestion of bovine food, as it takes very high temperatures to alter the conformation of the BSE prion, rendering it non-infectious. The current estimated odds of acquiring variant strain CJD from infected beef products is 1 in 10 billion servings. The majority of cases have occurred in Europe, with only 3 documented cases in the United States.
The infectious agent active in causing variant strain CJD is the bovine spongiform encephalopathy agent. This prion is responsible for the world-famous outbreaks of mad cow disease that ravaged much of the United Kingdom's livestock in the mid-2000s. Mad cow disease, or bovine spongiform encephalopathy, is caused by a prion like classic CJD, causing extensive damage to the central nervous system and the eventual death of cattle. The disease has a long incubation period, lasting up to 8 years, but exhibits a very rapid onset of symptoms once the disease becomes active. Cattle between the ages of 4 and 5 are most susceptible to the disease. Spread occurred in the United Kingdom due to healthy cattle being fed meat and bone meal containing brain and spinal cord tissue of infected cattle.
The symptoms of bovine spongiform encephalopathy begin with changes in attitude and activity of the cattle. The infected cattle begin to lose coordination and have trouble with simple movements. Weight drops and milk production dwindles in cows. After these symptoms are all in place, the infected beast eventually dies. Transmission is most common in consumption of central nervous system tissue, although the prion can be found in all tissues. Like in humans, the prion is able to avoid the cattle's immune system due to its protein form. Since prions are non-living, standard disinfection measures are useless against their spread. Only extreme heat or powerful chemicals are capable of denaturing the prion structure. The evidence presented for variant Creutzfeldt-Jacobs disease and bovine spongiform encephalopathy provide more than enough evidence to support a connection between the two diseases.
As they are both forms of transmissible spongiform encephalopathies and cause disease in both human and animal hosts, these diseases are strongly linked in their origin and virulence.
They are caused by prions, which disrupt normal protein folding, leading to degeneration of the host's central nervous system.
The fact that almost all cases of variant CJD have been identified in the United Kingdom, where the vast majority of BSE cases have been identified, also supports a strong connection between the two diseases.
Although the specific mechanisms of infection remain unknown to researchers even today, the overwhelming evidence indicates that these two diseases are nearly one and the same and can be said to be very closely related. Thank you.
Thank you.